Hello, my name is Jasmine Johnson. Today is January 20th, 2019, and today I will be demonstrating how to isolate individual colonies. So first, I'm going to show you, what I have here. I cleaned off this whole area with 10% bleach, and here is my alcohol with my inoculation loops. I actually have two of my inoculation loops in the bleach solution already because I already started off with two of my specimens and I've already collected them, but I want to go ahead and demonstrate the last one. So each one of these specimens, the first specimen is S. cerevisiae. It's cloudy. You can see some specimen or little spectacles inside of the actual solution, and that is the bacteria that grew inside. Same thing with the E. coli. The specimens are not as visible, but you can definitely see that the solution is cloudy. See some little tiny speckles in there actually floating around. And then the third specimen is the S. epidermidis, and this one is very small, cloudy too, and the specimens are a lot smaller, but I can see little tiny specimens floating around in there as well. So I'm going to show you guys how to demonstrate the aseptic technique with each one of these when doing the streaking inside of the agar. So first, I'm going to go ahead and grab my lighter and light my candle. The purpose of this candle is to create an open flame so that we can sterilize the tubes before we actually put the inoculation loop inside. So I'm going to go ahead and do my S. epidermidis. I'm going to go ahead and flame the end of the brim of the tube to make sure it's nice and sterilized. I'm going to grab my inoculation loop out of the alcohol. Make sure it's completely dry. Going to open up the lid of the agar, place my inoculation loop inside of the specimen while not touching the brim, and I'm going to go ahead and streak that first quadrant. Okay, then I'm going to place my inoculation loop back into the alcohol and close the agar. Then I'm going to go ahead and re-sterilize that tip, the opening, and recap. This will keep my specimen sterilized, and I repeat that step with each one of these. So I'm going to go ahead and go back to my inoculation loop out of the alcohol. That actually re-sterilized it by placing it in there. I'm going to reopen. The first quadrant I did was in this zigzag motion. I'm going to actually touch that quadrant and make my second quadrant in a zigzag motion. Then I'm going to make my third quadrant zigzag motion and my fourth one. Then I'm going to go ahead and close that up, and I'm going to dispose my last inoculation loop inside of my 10% bleach solution, and I will let that soak for 2 hours before I go ahead and dispose of this. And then with each one of my specimens, I will put this in the incubation area for 48 hours in order for the specimens to develop. And then I will store each one of my specimens in my test tubes and the refrigerator to use for further lab experiments. Hopefully you enjoyed my video and have a blessed day.